Hi, my name is Katelynn Horvath, and this is my summer at the University of Florida during the 2023 Chemical Engineering REU program. For context, I'm a rising junior at UConn. Last semester, I got involved in the ChEREU lab doing biochemical research. They are focused on the tau protein and the modifications of it common in neurodegenerative disease. I hope to assume a more active role in this research in the coming semesters. Accordingly, upon graduating, I hope to start a chemical engineering PhD program in this field. I took one of the first steps towards this goal by participating in the REU at UF. This summer, I worked in the Sampath lab with my faculty mentor Janani Sampath, my PhD mentor Yinhao Jia, and another REU student, Claire Cocker. The Sampath lab is a computational lab, and my work this summer centered around running molecular dynamic simulations, or MD simulations, using GROMACS. MD simulations are computer simulations of biomolecular systems used to predict the behavior of atoms and molecules over time, attempting to fill the gaps where experimental work is physically impossible or too expensive. MD simulations are particle-based simulations. Each particle is assigned an initial position, velocity, and potential. They are allowed to move according to these dynamic parameters, and at each time step, these parameters are recalculated. After many time steps, the simulation produces a final trajectory for the system used for data and visualization. To help define scale, I am running 100 nanosecond simulations, and each time step has a length of 2 femtoseconds. This comes out to 50 million time steps, meaning that the computer has to recalculate the position, velocity, and acceleration of around 300,000 atoms 50 million times. These robust calculations require UF's supercomputer, Hypergator, which enables data calculation and storage on an enormous scale. The basic steps of an MD simulation include generating topology, choosing the shape and size of a box, adding solvent to the system, adding ions to neutralize the system, performing energy minimization to ensure stable conformation, performing equilibration to make sure ions and solvent are equilibrated around the protein, and then running the simulation for data collection. Protein engineering is important in the world of protein therapeutics, which is diversifying every day and includes essential drugs like insulin. In my case, I am trying to engineer a CRISPR protein. These proteins are known for their genome editing and diagnostic abilities. The goal of this project is to find a way to make the protein BRCas12b, used for hep C diagnostics, more functional at higher temperatures. While already experimentally investigated, we hope to expand on it with simulations to improve the protein, prove the efficacy of MD simulations in these situations, and better understand BRCas12b's denaturation pathway. I was able to see protein denaturation using VMD to visualize the protein, but I also used numerical data like RMSD, number of hydrogen bonds, and hydrophobic solvent accessible surface area. Because the protein is relatively large for MD simulations at 1090 amino acids, I broke it down by its 12 functional domains. Separating the domains helped to identify the most flexible regions of the protein and start to see an unfolding pathway. Identifying these regions while also understanding their role in protein function helps us make informed decisions on how to modify the protein. In this project, I investigated adding a polyethylene glycol chain to the protein to stabilize it, which is called pegylation. This could be preferable to the prior method of mutations because it should not greatly affect protein function as long as the active site is avoided. As this project continues, we will have to consider many factors, such as molecular weight of peg, site of conjugation, and how these variables affect structure and function. I am so thankful for the REU program because of everything I have learned this summer, but also the connections that I have made. The REU program features social events on weekends with other REU students and the professional seminars where grad students and professors pass on their knowledge. I have gained so much perspective by being surrounded by such a diverse group of people that value engagement in the scientific community. Finally, a big thanks to everyone at UF who had a hand in making the summer great. Thank you.